DisProt curation training, annotating states, transitions, and functions of IDPs. Here are the learning objectives of this part of the training. Describe the types of structural states, give examples of the types of structural transitions, explain how to use gene ontology terms for additional annotation, and list the types of intrinsic disorder-derived functions. So let's start with defining the types of structural states for annotation in DisProt. In DisProt, you will be able to find 4 structural states for annotation: disorder, pre-molten globule, molten globule, order. So these are the 2 extremes. Disorder, it's a non-compact state without a stable 3D structure. Order, on the contrary, is a compact state with a stable 3D structure. There are also intermediate states that are in between these 2 extremes. We are talking about pre-molten globule and molten globule. Both have some structure, we refer to secondary structure. Both of them, though, are lacking specific tertiary structure, because pre-molten globule is a condensed state with residual secondary structure. Molten globule is a compact state with native secondary structure, but it's lacking specific tertiary structure. You might want to learn more about structural states of intrinsically disorderly proteins. You could do it by reading this publication linked here. Also, we just said that there are 4 possible states for IDPs, and you see them here. And you also see that there can be transitions from each one of these states to every other else of these states, meaning that we can have transitions from a more disorderly state to a more ordered state, transitions from a more ordered state to a more disordered state. The most common examples include disorder to order, and order to disorder. But we could also have other options. For example, disorder to pre-molten globule, it's still a transition to a more ordered state. You can see it here. And order to molten globule. So a transition to a more disordered state. And you can see it here, order to molten globule. Again, if you want to know more about the transitions of structural states of an IDP, you might want to read this publication that is the same that was linked in the previous slide. Then if we want to annotate the active functions of these IDPs, IDRs, we will use gene ontology terms. So we will use gene ontology terms to annotate molecular functions, biological processes, cellular components. And if you want to have a more in-depth understanding of terms, of gene ontology terms, you could look at these 2 resources that can help you a lot in understanding gene ontology and in performing efficient annotations, as well as look at the gene ontology website itself. All the gene ontology terms are available for search from the DisProt curation interface. So you will be able to add each gene ontology term from the curation interface. Again, you are required to provide the most specific term when possible. Other functions related to IDPs are functions that only pertain intrinsically disordered proteins and regions. These functions include, let's say, 3 main classes: entropic chain functions, molecular recognition display sites, self-regulatory activity. You're going to find these 3 classes of functions, as well as their child terms, available for annotation in DisProt. This is also the distribution of how these terms are currently annotated in the resource. Let's see now the entropic chain functions. You can have a flexible terminal tails, meaning flexible N-terminal or flexible C-terminal tail of a protein. Flexible linker spacer, which is also the most abundant disorder-specific term annotated as function in DisProt. What is a flexible linker spacer? It's a flexible unstructured region connecting two flanking domains that are structured. So the flexible linker is in between these two domains. When we have molecular recognition display sites, easily these are PTMs, post-translational modifications, that are found inside intrinsically disordered regions. Might be a glycosylated residue, phosphorylated, and so on. Then we have self-regulatory activity, meaning the binding of the intrinsically disordered region with another part of the protein to perform self-inhibition, self-activation, self-assembly. So this region is going to regulate